Okay, this is the third lesson to introductory course to high school level honors biology. And today we're going to talk about macromolecules. Macromolecules are essentially the blueprint of life. All living things, as we know, are built from these very large and complex molecules that are made mostly of carbon. And these molecules are essential for things like forming cells, storing energy, and transmitting genetic information. We characterize macromolecules in four categories, four types of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And most of which are what's known as polymers. And this means that they are made through linking smaller building blocks known as monomers, which I will explain in a second, into larger structures. And together, they carry almost every important function in any living organism. So most macromolecules are polymers, and the only exception are lipids. So the three others are polymers. A monomer is a single small molecule that can join with others to form a chain. And when they do that, we form what is known as a polymer, which is several monomers in a very, very long repeating sequence. And polymers are built through a process known as dehydration synthesis. Water is removed from a bond in order to join two separate molecules together. And we will see more of that down later in the slides. And polymers can be broken through hydrolysis, where now we add a water to break this polymer apart. Polymers are really easy to imagine in your head when you think of a necklace made of beads, which is how I like to imagine it. So on the right over here in the diagram, this very long bead-like necklace is known as a polymer. Different um, different macromolecules will have different monomers and thus different polymers, and we will get into that in a second. But I would like to remind you guys that this is generally how I like to picture it in my head. So the first one I want to talk about is carbohydrates. Carbohydrates are probably in the majority amount of foods you eat every day, like rice, potatoes, and they're made of carbon, hydrogen, oxygen in a 1-to-1 ratio. What this means is that the number of carbons, hydrogens, and oxygens in any form of carbohydrate, if you were to count them out and put them together in a ratio, it will all simplify to 1, 2, and 1. And most famously is the carbohydrate known as glucose. Glucose is the main source of energy throughout your body, and glucose's formula is C6H12O6. And if you were to divide 6, 12, 6 and simplify it, you will get 1, 2, 1. And carbohydrates monomers are what's known as monosaccharides. It's a very long word, but it means simple sugars like glucose, as I just said, as well as fructose that provide quick energy. Disaccharides in the name, di meaning two, are made up of two sugars. These include sucrose, lactose, and are used for energy transport. Polysaccharides, meaning multiple saccharides, are long chains like starch, cellulose, and glycogen used for energy storage and structure. Cellulose gives plant cells in the cell wall rigidity, while glycogen stores energy in animal liver and muscles. So then we move on to lipids. Lipids is the only macromolecule we will study that does not have a repeating sequence of monomers or individual identical, nearly identical units. Lipids are large molecules that are made mostly of carbon and hydrogen chains repeating over and over again, as you can see in the diagram on the right, and as I said, are not built from repeating monomers. So note that the carbon chains right here, each CH2 is not known as a monomer. A very important detail to note is that lipids are not hydrophilic. They do not like water. And if you try to mix any fatty substance, such as butter or like vegetable oil, into a glass of water, you will notice that it does not dissolve well in that solvent. Whereas if you mix something ionic, like salt, it will dissolve just fine. And these properties give it several uses, such as insulation and waterproofing, which is why in our bodies, we use what is known as trisulglycerides as our main form of fat, and they provide heat and insulation for our bodies. And right here is the trisulglyceride or triglyceride, another name for it, I've just mentioned. It's made of one glycerol molecule right here and three fatty acid chains, shown right here. Lipids can also be membranes, as well as serve other important functions. There are two types of fats that you need to know, first of which is known as the saturated fat. Saturated fats have straight chains and are solid at room temperature. So on the right diagram over here, you can see that every CH2 chain has a single bond between them. This means that they share the same constant 2 electrons between every carbon atom. So you can think of their relationship between each carbon molecule the same. Whereas unsaturated fats have sometimes what is called a bent chain, or more formally known as a kink. And this results from a double bond, shown right here. When we have a double bond, the literal shape of the molecule changes, and this is attributed to some of its properties, such as the fact that it's a liquid at room temperature. So vegetable oil is a very good example of an unsaturated fat. Next, we have phospholipids. Last lesson, we talked about the cell membrane, which use phospholipids as its main component, main building block. Phospholipids form the cell membrane with a water-loving head, as we have learned, hydrophilic head, and water-fearing tails, or as we have learned, hydrophobic tails. And through having water-loving heads and as well as water-repellent tails, we're able to form a cell membrane where we're able to regulate what comes across the cell membrane and what comes out. And lastly, we have what's known as steroids. Different from a little more general than the steroids you hear where in bodybuilding they're injecting these artificial hormones and they get all jacked up. Steroids in this case is a little more general term, and it includes cholesterol and hormones with specifically a certain amount of ring structures. You do not have to remember this, but steroids do include cholesterol and different kinds of hormones. And in the case of bodybuilding, the steroids we're referring to are artificially crafted hormones that are then introduced to the body. If you have any questions, just leave it in the chat and then, or just unmute and interrupt me. Next, we go to a very, very important macromolecule known as proteins. You've probably heard this term before. Proteins are certain substances made up of 21 different amino acids. In this case, amino acid is the monomer for the macromolecule. And not every protein has all 21, but usually I like to think of it like a Lego set where you have 21 kinds of different blocks. And with these 21 individual pieces of Lego, you can make all kinds of different creations. And as you can imagine, it's probably near infinite what you can do with it. You can put certain sequences that are different from one another. You can make different shapes. You can build different, I don't know, animals, planes, buildings. There's so many possibilities with 21 different kinds of Lego pieces. And the same can be said for literal proteins. These 21 amino acids can rearrange themselves in different sequences and form proteins that have very, very different shapes, properties, and functions. So I'll talk about the structures of proteins now. So the sequence of amino acids mainly decides the structure of the protein. And we have four structures that I will, yeah, okay, here. So we have four main structures. The first of which is the primary structure. This is just our polymer, where we have a multitude of amino acids linked together into a very, very long chain. The secondary structure is where we have interactions between certain monomers, between certain amino acids, known as more notably hydrogen bonds, as we have talked about in lesson one. Hydrogen bonds will cause the primary structure to begin folding into different forms and shapes, and we call these shapes either the beta pleated sheet or the alpha helix. And these are really easy to picture in your head. So imagine the beta pleated sheet as literally a sheet, and the alpha helix is a helical structure. Next, we get into the more functional forms of protein. So the things that actually do stuff in your body. And this starts from the tertiary protein structure. This is where we have all kinds of different interactions between the amino acids. The bonds that are formed gets really, really complex and goes beyond hydrogen bonding. And as a result, we have so many different possibilities of shapes and functions we can get from the tertiary protein structure. This is why it's one of the more mainly used structures in our body for whatever proteins need to be used for. And then we have the quaternary protein structure. This is an even more advanced form of tertiary, and it's also a functional form of protein. It's where we have more than one amino acid chain. So we have several of these polymers joined together to form one functional body. Remembering the structures of proteins is pretty important when we're going on later to talk about certain properties, understanding the bonds, and as well as the functional groups in proteins, and something that's known as denaturation. I'll check if it's might be back here actually. Um, where did I write denaturation? Here. Anyhow, denaturation happens when certain here. So certain temperature changes or pH changes can disrupt the structure of the protein. This is one of the reasons why your blood has a certain acidity or basic level known as pH, as well as why you have to maintain a constant or around constant temperature within your body. Your proteins, most probably every single one of them, will do something known as denature when your conditions within your body get too extreme. And this happens when the bonds that are holding the amino acids together between different amino acids that form things like these very complex structures. So imagine like magnets between them, and you just magnet stick together in different ways, and you just make like a mess. Begin unraveling because the bonds can no longer hold themselves together. And when they unravel, we go back to these more simple structures. And what this does is that essentially the protein just loses its function. This is why it's really important to maintain a certain pH level within our bodies as well as temperature. So I'll talk about each individual amino acid now. On the right, this is the basic fundamental format for an amino acid, and it is made up of four, I like to think of it, groups. The first of which is known as the amino group right here. It is made up of one nitrogen and two hydrogen atoms, or NH2. And the next one is the carboxyl group, which is one carbon atom, two oxygen atoms, and one hydrogen atom. These two groups are more generally and largely known as functional groups, where if you see these molecules, NH2 or CHO2, in other molecules of any kind, we will also identify them as an amino group or carboxyl group. Next, we have a hydrogen attached to the center carbon. And here comes the most important part. We have something that's called an R group attached to the center carbon. This R group acts as a variable. It is different between every single 21 of the amino acids. So every like 21 amino acids, every single one of them has an amine group, that hydrogen, that carboxyl group. The only thing that's different is going to be the R group. The R group can be close to anything. The most simple amino acid has, I'm pretty sure, just a hydrogen for the R group, which is called glycine. So it's just the hydrogen here. But some of them can get very, very, very complex. So they branch out. And what this does is that the molecules in the R group have certain properties that allow the amino acid to interact with other amino acids. So I like to think of it like if you had a bunch of straps, um, think of it like those shoe straps that have Velcros on them. If every single strap had Velcros in different places, so maybe some of them have all the Velcro attachment on top, and then it's just like nothing at the bottom, it's just the strap. And if I have another one where I have like maybe diagonals, maybe different ones coming across, and I try to stick them together, and when I loosen it like this, every single one of them is going to look different, right? Because we have different molecules that like to attract to other molecules, or maybe some molecules that like to repel from each other. So imagine these Velcros now as amino acids. When we put amino acids close to one another, these Velcros or the R groups begin interacting, and it forms a very either strong bond or repelling, and this affects the general shape of the protein. The bond between every single amino acid that links them together to become a polymer is known as a peptide bond. So as you see right here in the bottom, the carboxyl group has given up a oxygen and a hydrogen, and the amine group has given up a hydrogen. And if we go back earlier where we talked about dehydration synthesis, we will remove a water molecule to join two monomers together. So if you see here, the one oxygen and the one hydrogen lost added on to the one hydrogen lost from nitrogen conveniently becomes H2O. So after we've removed H2O, the carbon can now successfully bond with the nitrogen. And like that, we link the amino acids together. And note that a chain is not a protein yet. It is what's called as a polypeptide. And eventually, later on, after many adjustments, um, eventually folds and becomes a functional working protein. And we'll talk about that when we get to what's known as translation in DNA. So now let's talk about nucleic acids. Nucleic acids include DNA and RNA, and they mainly function as the genetic instruction within our bodies. So I like to think of it as the blueprint, the map, the instructions, the hand manual for everything that happens within your body. And for simplicity reasons, I will talk about DNA and RNA within our cells. Just note that some forms of DNA and RNA within viruses or other organisms are a little different than the ones we see within our bodies. For example, DNA can sometimes be single-stranded, as well as RNA can sometimes be double-stranded. So I'll explain that in a second. But for simplicity, we're only talking about the ones in our body. But just keep your mind open that these molecules can vary when we get into more complicated organisms like viruses. So nucleic acids are made up of, no exception, monomers, but the more formal term for these monomers is called nucleotides. Every nucleotide is made up of a sugar, as you see right here, a phosphate group, as you can see right here, and what we call a nitrogenous base. The nitrogenous base is a part of the nucleotide that changes, that is variable. Similar to the amino acids R group, the nitrogenous base has a selection between four different nitrogenous bases that give it different properties. And these bases within DNA are adenine or A, thymine as T, cytosine as C, and guanine as G. And RNA has thymine. RNA replaces thymine with uracil, which we denote as U. DNA structure is we take these building blocks, imagine a nucleotide, and we stack them onto each other to form one entire strand. And now we take two of these strands and we form a double helical structure. That is known as DNA. RNA, however, is just one strand. And the main difference is, apart from being single-stranded, is you have to remember that RNA does not have thymine as a nitrogenous base. It only has uracil. And the sequence of these nitrogenous bases or nucleotides, when we're putting them together in a strand, is essentially the core of all life. The varying differences between each strand, the specific sequences that we're writing, is the code. It's literally the genetic code for every process within our human body. When, let's say, let's say like there's a growing fetus, when it wants to grow, like, let's say an arm, or it wants to grow these hands, these fingers, it reads the sequence So let's say adenine, cytosine, cytosine, guanine, thymine, 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 adenine. It reads it from this strand at a super speed, and then from that, it knows the information as well as the instruction to create this hand and these fingers. And it's pretty crazy to think about because every single thing we're able to do, every process, millions, billions of things happening are all attributed to these otherwise seemingly simple sequences, microscopic sequences within our body. And we also have to learn something called central dogma. Central dogma is we derive RNA from DNA. So I'll explain it. Imagine the nucleus within your cell, the nucleus, the ball with all your DNA, and the DNA gets transcribed into RNA. So when we're delivering the instruction manual, when we want to tell your body what to do, we're not taking the actual DNA because the DNA you can't expend this. You can't spend it because if you keep using it, the cell no longer has DNA. So instead, it creates something close to a copy known as then the RNA. The RNA is what now is able to deliver the message of what the DNA wants to say. So the DNA doesn't do it itself. It doesn't really leave the nucleus. The RNA does. And RNA, if I, no, I didn't. RNA, apart from being a messenger, can also serve several different functions. So the messenger form of RNA is known as mRNA. It helps send the message from DNA into then your ribosomes and your other cells. Um, oh. And the other forms are tRNA as well as rRNA. So tRNA is the RNA that comes during a process known as transcription. Translation. Translational RNA. And it's the one that helps attach the amino acids. So I will explain that next lesson. And rRNA, which is meaning ribosomal RNA, where RNA is sometimes used as, I guess, a building block, a construction tool for ribosomes. And because of this, we used to actually think that RNA was the main source of genetic information within our bodies. And some point in this timeline, it has switched to DNA. So let's talk about all the experiments leading up to what we know about DNA today. The most important experiments, which are three, that you will pretty much always learn in every single biology textbook. I promise you, if you go to any library, any store, and you flip open a biology textbook and go to a certain chapter, you will find the exact three experiments. The first of which is Griffith's transformation experiment, which happened in 1928. Frederick Griffith was studying two types of bacteria that caused a disease known as pneumonia in mice. One strain was smooth and deadly, and one other one was a rough strain, but it was non-virulent, meaning it was harmless. And when he had injected these heat-killed smooth strain into mice, the mice lived. But however, when he injected the heat-killed ones with seemingly harmless ones, the mouse still somehow died. So let's look at it again. Inject a harmless strain, the mouse lives, that's simple. Inject a virulent strain, the harmful one, the mouse dies. Simple. Inject one where we have killed the virulent cells, the mouse lives. Simple, because the virulent cells can no longer do their job, and the mouse can then live on. But if we mixed these two together, these two cases where the mouse lived, somehow the mouse died. And Griffith noticed this. And he said that something from the dead bacteria that was injected into the mice transformed the non-virulent strain into a virulent strain. So some interaction happened between these two strains. And this is the first evidence ever that, or well, experimental evidence, sorry, that genetic information could be transferred between cells. And this started a series of experiments where we wanted to know what that genetic information is. So at that time, we didn't know it was DNA. It was just starting the race to what the heck was transmitting this information between these strains and bacteria. So now we come to Avery's experiment. Avery and his fellow colleagues wanted to know what that transforming material that Griffith referred to was. And they repeated this same exact mice experiment, but each run, each trial, they removed a different molecule from the dead bacteria. So they wanted to prove that, for example, if we removed lipids from the dead bacteria and the dead bacteria no longer transmitted the virulent DNA, sorry, not DNA, information, then we can reasonably conclude that lipid was the transmitter of this information, because when we destroyed it, the mice didn't die. So they destroyed certain molecules every trial, and all the way until they came to DNA. And when they destroyed DNA, the transformation stopped and the mice did not die. So they concluded that DNA was the molecule responsible for passing on genetic traits. But it doesn't end there. Almost 10 years later, there was still a little debate, confusion between whether proteins or DNA was the actual transmitter of genetic information or holder of genetic information. And that's when Hershey and Chase came in 1952. And they worked with something known as bacteriophages, viruses that are able to infect bacteria. And what is known about these is that they tend to inject their own genetic material into a cell. So what these guys did was that they traced the protein with radioactive sulfur and the DNA with radioactive phosphorus. So let's go back to protein. So let's be clear about why they did that. Sulfur and phosphorus are exclusive to proteins and DNA. One does not have the other. DNA does not have sulfur, and protein does not have phosphorus. So let's take a look. In some proteins, or in a lot actually, the R group contains sulfur. And if we look down to nucleic acids, this right here is the phosphorus we're talking about. This is what we're tracing. So if we put tags, if we put labels onto both of these molecules, and we allow the bacteriophage to, right here, inject or infect the cell with its genetic information, we can then look at the cell and tell whether the radiation was coming from sulfur or was it coming from phosphorus. So if we label protein and we allow the infection to happen, and there was no sulfur record in the cell, then protein cannot be the genetic information. Whereas when they allowed the bacteriophage to infect it with a traced phosphorus, we then picked up after centrifugation, so we spin it really, really fast. We then picked up phosphorus within the cells. And this was definite evidence that DNA was what was transmitting the cell's information. Oh, and I forgot to add one really important one. I think later in one year, I think 1953, Rosalind Franklin was the one that used X-ray crystallography to then determine the helical structure of DNA. I thought I could put this in next lesson, but it feels pretty important to talk about now. And then Watson and Crick were the ones that published the work and established the structure of DNA. And this allowed us to look into things like the amino acids, as well as certain other properties of DNA, all the way until what we know today as the macromolecule. So there is a lot to talk about DNA. You might feel like I have already spoke a lot about it, but trust me, there's more. DNA is mainly involved in two processes, which is transcription and replication. So how does DNA actually replicate? We can't just have a set amount of DNA and just leave it there. Like eventually, you're gonna need more. Your cells are constantly dividing. You always need new DNA for every single new individual cell, as well as transcription. So how do we actually get the information from the DNA? You can't just say it's a bunch of like molecules that are a little different from